Okay, so we will continue talking about vitamin A. This second slide presentation for vitamin A focuses on the various functions of vitamin A. So versus reviewing the more complex metabolism of vitamin A in the enterocyte and hepatocyte, we'll be talking about the specific functions of this nutrient. So we see that the retinoid and the retinoic acid forms are involved in vision and gene expression. We also discussed the role of carotenoids as an antioxidant. The presentation concludes by outlining the DRIs for vitamin A as well as discussing its deficiency symptoms.
We can see here the number of roles that vitamin A plays in the body. So we see that it's involved in vision, especially in the dark, cellular differentiation, gene expression, growth, other functions involving reproduction and bone metabolism, and we'll also see vitamin A and its antioxidant role. First, we'll discuss vitamin A's role in vision, especially in the dark. And so we see here a diagram of the eye. We see specifically in further detail the outline of the retina, which contains the rod cells and the cone cells. And we see here this rhodopsin is a component of the rod cells. So we see the rod cells, and then we see rhodopsin as being a component of this. And we see that rhodopsin is made up of opsin and then what is called cis-retinol. So cis-retinol is from vitamin A. It's derived from one of the retinoids. So we see this is from vitamin A and then opsin, and then these bound together will form rhodopsin. So we'll see here the process of the exposure to light and then what occurs with this rhodopsin and how we have this being involved in vision. So we see first here when the light hits the retina in the back of the eye, what happens is the rhodopsin in the rod cells are actually transformed and signals are sent to the brain. So rhodopsin's transformation involves its cleavage. So when we have the light that hits to the back of the eye, that affects the structure of rhodopsin and actually this bond here that is formed between opsin and cis-retinol. So we see after that exposure to light, that bond is broken. So we see that there's a cleavage of rhodopsin and we see release of opsin and cis-retinol. And additionally, what you see with this exposure to the light, that the cis-retinol is converted to trans-retinol. What will need to occur next is the reformation of rhodopsin for this process to occur again. But for the reformation of rhodopsin, the trans-retinol will need to be converted back to the cis form. So we'll need to convert the trans back to the cis retinol, and then when converted back to the cis retinol, then it can join with opsin to again form rhodopsin, and then we can go about having that exposure to light, and this is all going to be involved with the process of vision. So we can see here the steps again involved with vision. So we see that vitamin A in the form of the cis-retinol is needed to form rhodopsin. And we see that rhodopsin is made up of the 11 cis-retinol and opsin. So these are bound together to form rhodopsin. In the dark, when light hits the retina, the rhodopsin is cleaved, releasing the opsin and the cis-retinol. The cis-retinol at the same time is also converted to trans-retinol. So we see here to regain vision, the rhodopsin compound must be resynthesized. So for that to occur, that trans-retinol is converted back to cis-retinol, and when it's converted back to cis-retinol, it can be recombined to join with the opsin and then form rhodopsin again. So when it's converted back to the cis-retinol, it is transported back to the rod cells, it combines with opsin, and then can form rhodopsin. So we see here related to this process that vitamin A deficiency, and with the vitamin A deficiency, we're forming less of this cis-retinol, which can affect rhodopsin formation. So we see with the vitamin A deficiency that can contribute to what is called night blindness. So you can review again this diagram and the process of the cleaving of rhodopsin, and then the conversion of cis to transretinol, the conversion of transretinol back to cisretinol, and the reformation of rhodopsin. Next, we will discuss the function of vitamin A binding to its nuclear receptor and then affecting gene expression. So an important component of this process is the retinoid that is involved. So the specific form that's involved is the retinoic acid form. So the retinoic acid form is the form of vitamin A that binds to the nuclear receptors, and it's binding to the nuclear receptor that is attached to what is called the vitamin A response element. And so we'll see a diagram of this process of the retinoic acid binding to its nuclear receptor. Another important component of this process is this compound here called CRABP. It stands for cellular retinoic acid binding protein, or the CRABP. So this compound actually binds to the retinoic acid and shuttles it into the nucleus so that it can bind to its nuclear receptor. And we're going to review this diagram. We see that these two different forms of retinoic acid, either the all-trans retinoic acid or the 9-cis retinoic acid, so these two different forms of retinoic acid will bind to a specific receptor. So we see that the all-trans retinoic acid binds and activates the RAR, which stands for retinoic acid receptor, RAR, and then this 9-cis retinoic acid form binds and activates what's called the RXR, which stands for the retinoid X receptor. So you want to link here the all-trans retinoic acid to RAR and the 9-cis retinoic acid to the RXR. We'll see also in the diagram if vitamin A is not present. So if either of these retinoic acids are not present, then the RAR and RXR receptors will bind to what's called a co-repressor, which inhibits transcription. So we're going to see that these retinoic acids will promote transcription, but when the retinoic acid is not present, then the receptor binds to the co-repressor, inhibiting transcription. So we see here a diagram of the process. We see the different forms of retinoic acid, the all-trans retinoic acid and the 9-cis retinoic acid. So they're going to both be bound to the cellular retinoic acid binding protein. So we see here the CRABP bound to the retinoic acids, and the CRABP will shuttle the retinoic acid into the nucleus so that they can bind to their nuclear receptor. So the all-trans will bind to the RAR, the 9-cis will bind to the RXR, and these two vitamin A receptors form what's called a dimer. So a dimer is a combination of two different receptors bound together. So we see the dimer here, and the dimer is also attached to this response element, the retinoic acid response element, or RARE. With the binding of retinoic acid to the nuclear receptor, that will actually affect the mRNA transcription and then protein synthesis. So it's going to affect the gene expression, it's going to affect mRNA transcription, and then it's going to affect protein synthesis. And these effects is going to be involved in the role of vitamin A in cell differentiation, cell proliferation, in growth, and other effects of vitamin A that we're going to see in some of these additional slides. So we see additional functions and mechanisms of vitamin A. So vitamin A as retinoic acid is needed for cell differentiation. We see it promotes differentiation of keratinocytes to mature epidermal cells. Also, the retinoic acid is thought to regulate proliferation and differentiation of the myeloid precursor cells in bone marrow, which is involved in immunity, so immune system function. We also see a role of vitamin A in growth. So with vitamin A deficiency, that's associated with impaired growth. The exact mechanism is not known specifically, but we do see a link between vitamin A deficiency and impaired growth. It is interesting that the effect of vitamin A is cell specific. So in some cells, it actually promotes cell proliferation. In other cells, it promotes cell differentiation or cell death. So the effect that it has is not consistent, it's cell dependent. Some is going to be the proliferation and some cell differentiation. This is going to be occurring in part through its ability to bind the nuclear receptor, affecting gene transcription and protein synthesis. Additionally, retinoic acid increases the synthesis of what are called gap junction proteins, which promotes cell communication. Next, we see a role of vitamin A in bone development, in hematopoiesis and iron distribution, as well as immune system function. So with bone development, what is interesting is that vitamin A that is either too high or too low, so vitamin A status that is either too high or too low, can negatively affect bones. So you want to make sure to have enough, but not too much or not too little vitamin A. Also with the hematopoiesis and iron distribution, it is recognized that those with low vitamin A status can actually exhibit symptoms of anemia. And also we see a role in immune system function, so those vitamin A deficient have an impaired ability to fight infection. We'll now discuss the antioxidant functions and antioxidant roles of vitamin A, and this is specifically through the carotenoids. So a lot of these previous effects that we've discussed, so vitamin A and its role in vision, binding to nuclear receptor, affecting growth, affecting bone, affecting these other tissues and processes, has been specifically related to retinoids, but we see that the antioxidant role of vitamin A is through the carotenoids. So we see as antioxidants, carotenoids being involved in eye health, specifically lutein and zeaxanthin. We see these are found in the macula of the retina, inhibiting oxidation of cell membranes. Also, we see a role of carotenoids in reducing risk of heart disease. We see that it prevents the oxidation of LDL cholesterol, thus slowing the process of atherosclerosis. We will now discuss the link between carotenoids and cancer, and theoretically the carotenoids' antioxidant mechanisms may appear to be cancer protecting. However, intervention trials in humans have not shown a beneficial effect of vitamin A or beta carotene in preventing cancer. One study actually showed an increased risk of lung cancer among smokers in the supplemented group, so the groups supplementing vitamin A and beta carotene. So the study again showed an increased risk of lung cancer in the supplemented group versus a placebo group. Therefore, the Institute of Medicine does not recommend beta carotene supplements for the general population. We will now review the RDA for vitamin A. So we see for men it's 900 micrograms per day, for women it's 700 micrograms per day, and we see the tolerable upper intake level being 3,000 micrograms per day. So then we see this conversion, so 1 retinoic acid equivalent equals 1 microgram of retinol, and also with 1 microgram of retinol equaling 12 micrograms of beta carotene. So we see that beta carotene has higher conversion compared to the other carotenoids, compared to alpha carotene or the beta cryptozanthin. We'll now discuss the deficiency symptoms and conditions associated with low vitamin A status. So we see what's called xerophthalmia, the keratinization of the cornea. We see that with the keratinization that cells lose water content, so it dries out the eye, filling the eye with this keratin protein, and so with extreme cases the eye can become very, very dry. It can almost have a leathery appearance, and it can actually lead to blindness in severe cases. With toxicity, it's called hypervitaminosis A, and this is going to be primarily with high supplementation of vitamin A. So in supplement form, it can cause nausea, vomiting, and headache. It can be teratogenic, which is actually harmful effects to a developing fetus, and it can negatively affect liver function.